this is Chris Haugen, Brussels Scientific Institute for Public Health.
So in order to facilitate that we get the most of the people there engaged on the webinar, I would encourage to invite people, other applicant groups that you think they might be interested, so we get more questions even than today and more interest from people. I mean, it's been very short notice and I miss, a lot of people told me they couldn't come, no?
I think...
Yeah. Yes, Martin, you had that slide on the expected in-kind contributions of the EFPIA member companies. These are in some way also competencies.
Mm-hmm.
Does it mean that in the expression of interest, they don't need to be addressed or need to be taken as such?
The expressions of interest shouldn't be addressed, you mean?
I mean in the expected contributions of the FPA members. So does it mean that that should be taken for granted, that these technologies will be used by these companies and that they don't need to be addressed by new small companies or other partner organizations?
You want to take it? I complement afterwards, yeah.
No. In fact, as Martin mentioned, what all the companies are looking is for new ideas. So we know perfectly our models and the way we run, but if someone comes with a good new model or an improvement of a known model, this will be considered.
I mean, I insisted a lot that the ultimate goal is to integrate, and we really want to innovate in how we optimize, how we transition regimens, not just drugs, not just single agents, even regimens. But saying that doesn't mean we don't want more models. But we don't want, I mean, we would prefer an expression of interest. I think it will help more on achieving the overall objective. That is not just coming with all sorts of new models and let's see if they deliver or not. It's more about integrating the information we have and complementing the information that is not already generated by the current models. So we do a better transition, improving in the mathematical modeling, improving on how we predict efficacious exposures, how we integrate safety also.
Can you hear me?
Yes.
In France. I think one of the major bottlenecks is novel compounds. And I think we saw, at least from your presentation, only GSK is providing a library. Are there other inputs coming? Because I think that's the first and most important is new compounds.
Yes.
As models, I think we have sufficient. And if you want to develop new compounds in zebrafish, welcome. But I think this is more a scientific approach, not really directly related. And the second question I wanted to ask is how many projects, if they are excellent, will be considered? One. So we are fighting always with each other.
Well, if you complement each other, you can always apply as many members as possible, no, in one consortium, but you have to be in agreement, no? It's true that the main bottleneck is the lack of compounds, the lack of good targets. The topic, it doesn't go about that, but we need ideally to get these compounds into the topic, into this project. GSK is willing to do it. Other companies might be reluctant because of IP. But this is something that we cannot address at this point. I think once we have the applicant consortium on board and companies see exactly what is going to happen here and what kind of work we are doing, I presume they will see the big advantage of putting their compounds through this project to be assessed and really ascertain the information they cannot do by themselves. This is something to take into account, to work on it, but until February is difficult to go further than where we are.
Can I also just make a comment here? Yes, as I say, I mean, the IMI project, since a project where you have several companies working together, are deliberately staying away from development of new compound, which is something that is completely impossible for companies to work together. But which doesn't mean that, I mean, here we are talking about the fact that what is really bottleneck is to be able to elaborate, to find some regimen that can be worked out in animal models so that then you can then go and test in humans and get to your efficacy stabilized in a more shorter and more secure way, which doesn't mean that maybe company could come with compounds that are already sort of clear up and used in a certain way and already maybe even out of the patient, and then test it in an innovative way using the input that will come from the academic, from the SME consortium, and get to something new.
In a worst case scenario, let's say we only work with current drugs, still there's a lot to do. We don't know well how they work. We have to innovate in how we use combinations. We have to innovate in how we guide early clinical development trials. And then we are in conversations with the Critical Path Initiative, no? And they have a drug coalition. So most likely they will be providing drugs through that drug coalition. I don't know how many, and data sets. And then TB Alliance also will play a role at a certain point, but we have to wait until we get the merge of both consortiums until we open up all those discussions. Yes.
Microphone to the back,
Malina from Accentum, Switzerland. I noticed that all the drug used now for tuberculosis, a chemical entity. Even I contact tuberculosis alliances with possibility to develop the immunobooster. They said, no, we are not interested. We are working just on small molecule. And I think that we can do something better. We can make immunobooster which stimulate the immunity of the patients and clear from tuberculosis these patients. Mm-hmm. And when we use small molecule, it is very well known that cigarette smoking leads to disease. You used small molecule, as in cigarette smoking. That means that you modified your proteins, and you will have the side effect. The side effect, perhaps, the tuberculosis, you will kill the bacteria, but you will have the tissue degeneration, the lung degeneration, which you couldn't stop by any small molecule, because small molecule lead to side effect and lead to degeneration in general. Mm-hmm. That's why we have this epidemiology: don't smoke cigarette, but take the drug which will kill you. We should change. We should change. What is the flexibility of EME to accept new story? That means in vivo model, you accept in vivo model for small molecule. It will be completely another model for my immunobooster. And I can stop tuberculosis by using immunobooster to misfolded protein. You will create by the small molecule misfolded protein. That means that the model in vivo could not be the same at all. Is some opening for completely new idea to stop infection? Thank you.
I think we are open to it. Initially, this topic has been written having in mind small molecules, but certainly there is room for biologics, there is room even for vaccines. We are in talks with different parts of the company, of the companies who already belong to the EFPIA consortium, about bringing the vaccine people on board. I mean, the minimal contribution they could do is exploring immunity, human granuloma, ex vivo bactericidal studies. But certainly we can expand in that direction if it helps on the drug development. So we are open.
very much. Joaquin, Trinity College. So I just want to start with fight with burn rifle and say that I think there's lots of room for new models. I mean, I think if all you needed was to get your drugs tried on the existing models, you probably wouldn't be here. And I think that there are a lot of exciting new models in academia which may get some oxygen from IMI, and I really hope that that comes through, like bioluminescence of mice, like new techniques for even estimating CFU with BAC-T devices, small changes and big changes. I run a TB cell biology immunology group, and we made our name with the TNF blocker story. I'm also a TB doctor, and we're getting ex vivo models from bronchoscopy patients and who are taking TNF blockers or who are smoking and who have got very perturbed cell-mediated and macrophage responses. And we think these things are germane to what you're trying to do, which is make a difference to the patient. So I think these ex vivo models are not available to you because you don't have a license to bronchoscope like I do. But I think that they do have a contribution. And taken in some, and hopefully not fighting each other, but recruiting a lot of people here who can do the other aspects, I think there's a lot to contribute here.
Yes, that sounds excellent. This is the kind of things we are looking for.
I have two questions about potential partners. Sorry, Greg Dow from Liverpool School of Tropical Medicine. What's the approach of IMI to partners from the United States? I assume they can't receive funding, but can they be part of the consortium if it's appropriate? And also, what should our approach be if we're already working closely with people from GSK, for instance? Should they not be involved in the expression of interest because we know that they'll be brought in as part of EFPIA?
Well, to the first question, is you are absolutely right. So for being eligible to funding, you need to be Europe-based, like for the majority of also actually FP7 program, apart from particular exception. So yes, for funding, you need to be in Europe, but that doesn't mean that you cannot participate. So everybody can participate, but you might not be eligible for getting money from IMI. So this is the first point. Then the second point is, well, GSK is already in the topic, in the consortium, because it's part of the EFPIA consortium, so of course they cannot participate twice.
Yeah, and I presume a lot of academic groups in the US somehow will contribute. It's not going to be matching, but we don't want to stop anybody from contributing. So we have also collaborations there, and all the information that we are getting in the company, we will try to put it through. Maybe the other companies will do it. And CPTR certainly, are expressing they are willing to collaborate. A lot of data will be shared there in CPTR. CPTR is mainly about regulatory science development, no? And it's going to be probably more clinical than preclinical. But there has to be a dialogue there. It doesn't make sense that we work in one direction and they work in another direction, and there is no, because we aim to translate all what we're doing.